All right, I think we'll go ahead and get started. Thank you all for coming today. Really excited for you to hear from today's speaker. Having spent some time with him, he's not only done some really amazing things, but as you're going to see, has just an infectious enthusiasm for taking on big challenges, which is really awesome and just incredibly accomplished with everything he's done. So Sean McClain, CEO and founder of Absci, based just up the road in Vancouver, Washington, is our speaker today. Absci, which Sean will tell you more about, is a generative AI drug creation company. So if you're like me, you're looking like, all right, where do I invest in this thing? That sounds good. And the mission is to create better biologics for patients faster. Sean really embodies the entrepreneurial spirit that we like to showcase in this series. Many of you come back for all these. We've had some really fantastic speakers. And Sean is going to be among those, I have no doubt. He built Absci from a basement lab in 2011 to a publicly traded company, so that's amazing. Scaling the company today to 160 employees, which he calls unlimiters. Haven't had that conversation yet, so hopefully you'll tell us what you mean by that. And they have expertise in AI, immunology, synthetic biology, and protein expression. So skills that many of our graduate students are developing here for the research that you guys are doing in the labs. Sean has raised over $525 million in capital and went through an initial public offering for Absci. He's established a growing number of industry-leading partnerships, which you'll see are key to their business model, and built an integrated drug creation platform that merges bleeding-edge generative AI with proprietary high-throughput wet lab data. Using all that, now Absci generates 3 billion high-quality, high-throughput protein interactions on a weekly basis. So that's just amazing. So a little bit about Sean. Sean studied biochemistry and molecular biology at the University of Arizona. He's been the recipient of many, many awards. I'm not going to go through all those, Sean, just in the interest of time, for all his work in innovation and entrepreneurship. One of the things he does here in Oregon, though, is he serves on the board of the Oregon Translational Research and Development Institute, or TRADI, and also Life Science Washington. Now, we're going to do something a little bit different today. It's just a slight twist from what we normally do. Sean's going to speak for about 30 minutes about his journey and what Absci does, and then we're going to have a little bit of an extended period for discussion. So I encourage you to think about your questions, build up some questions, and we'll start a really interactive Q&A period after Sean makes his comments. So with that, please join me in welcoming Sean McClain.
Thank you. Thank you, Bob, and thank you for the University of Oregon for hosting me here today. I disappointed my dad by not coming to the University of Oregon. Both my great-grandfather, my grandfather, and my dad were all Duck alums, and I decided to become a Wildcat. So, but I am a Duck at heart, so it is great to be here, and it's amazing all of the work you all are doing here, especially at the Knight campus. And before diving in, I'd love to play a little video that really demonstrates and highlights the passion we have at Absci for getting better drugs to patients faster.
There are thousands of life-saving therapies waiting to be discovered. But finding each one is like looking for a needle in a haystack. Meanwhile, patients and loved ones wait, hoping for a miracle before it's too late. What if we stopped looking for needles and started making them? Creating medicines we could never find because the antibodies don't yet exist. We'd need high-quality data, lots of it, and artificial intelligence built specifically to design these new antibodies, and a team of brilliant scientists to validate the designs in a state-of-the-art wet lab. We'd create new medicines in months instead of years, and could eventually design medicines for patients with the click of a button. This isn't science fiction. We are the company unlocking the design of new antibodies on a computer from scratch. And we're just getting started. With generative AI, we hope to be able to design more effective therapies that reach our loved ones faster, driving the future of drug creation. Because those who need life-saving therapies don't have time to wait. We are Absci, creating drugs at the speed of AI.
I get goosebumps every time I watch that video because it really embodies the passion we have at Absci of getting better drugs to patients faster than ever before, and the passion we have to really change how drug discovery is done. This broken process takes over 5.5 years just to get a drug into the clinic, another 10 years to actually get the drug approved. Now, billions of dollars are invested just to get one therapy to patients. And the success rate, extremely low. Less than 5% of drugs actually make it to patients at the end of the day. And it's because we have this trial and error process in drug discovery. It's like finding a needle in the haystack. And what we've done here at Absci is really flip that paradigm on its head, and using our scalable wet lab technology with generative AI to really go from searching for a needle in the haystack, this trial and error process, to actually creating the needle, in our case, an antibody with the attributes that you want the first go around, ensuring that you have the efficacy, the developability, the manufacturability. And this is what we see as hugely critical for changing this paradigm, being able to dramatically reduce the time it takes to get these therapies to patients, being able to increase the probability of success, unlocking new biology. And there's been a lot of hype that's been out there, but what we're showing here at Absci is that this is actually a reality. We've actually taken this technology and applied it to our own assets. One in particular that I'm going to be talking about today is TL1A. This is a target for inflammatory bowel disease. And what we were able to demonstrate with that is that we can use generative AI to dramatically reduce the time it takes to get into the clinic. So normally it's 5.5 years. With this particular asset, we'll be in the clinic within 18 to 24 months. Additionally, we are designing this in a way to achieve best-in-class attributes. We're increasing overall half-life, actually looking to go after patient populations that currently aren't being addressed, and so unlocking kind of new novel biology. And we're doing this at a fraction of the cost. Normally, just to get one asset into the clinic is $50 to $100 million. And with this particular asset, we will be in the clinic for roughly $13 to $15 million. And so you can think of the multiplier effect that this has. So instead of investing in just one asset, you can now start investing in five to seven different assets, in addition to increasing that overall probability of success. Now, we're here to talk about the entrepreneurial journey. We didn't start off as a AI drug discovery company. We actually started looking to see how we could engineer a very simple organism, E. coli, to produce antibodies. And like all great entrepreneurial stories, it started in a basement lab. I was at the University of Arizona, really passionate about how we could be applying engineering principles to biology, and really got obsessed with how the industry started with Genentech. Genentech's first drug that they had created was insulin. They produced it in E. coli. Then the industry shifted to monoclonal antibodies. And Genentech actually tried to produce an antibody in E. coli, and it just wasn't capable. And by switching from a simple organism like E. coli to mammalian cells, development times went up, manufacturing costs went up. And it was this really simple idea. If we could engineer E. coli to be more mammalian-like to produce these antibodies, we could really change how biomanufacturing is done, reducing the overall timelines and the overall costs. And so I ended up graduating a year early, moved back in with my parents, and I'll talk a little bit about some of my mentors later. But one of the mentors I had was actually my dad. He was the one that really encouraged me to go pursue this idea. I was actually planning on going to go get my MD, PhD. And I was talking to my dad. I was like, Dad, I really do believe that we can change how biomanufacturing is done, and we could make this happen. And he's like, Sean, go do that. And we sat around a kitchen table and figured out all of the used, surplus lab equipment that we could buy, pennies on the dollar. We, ended up finding a basement lab in downtown Portland. That literally, it was a 200 square foot basement lab that had wooden lab benches all across the wall. It literally looked like it was built from, Home Depot, and you had a garage sink. And I remember, putting in all the equipment, and I mean, it literally looked like a dungeon. And my dad's, helping, move the lab equipment in. And he's like, that's when he started kind of questioning, like, was this a great idea? He's like, are you sure you want to do this? And I was like, absolutely. It was the passion. It was the passion that I, believe that we could, change some of these fundamental problems within the industry. And so I started doing all the genetic engineering, showing that you could actually produce these antibodies in E. coli, and completely naive to the industry, had no industry connections whatsoever, and I knew that I needed to go get some initial partners, some initial customers, and so I reached out to the top 100 large pharma, and I had no email addresses, no contacts. So I literally was going, and searching for the generic BD email addresses at each large pharma. It was like bd@lilly.com. Hi, I'm Sean McClain, looking to, change how biomanufacturing is done. Would love to chat with you about, how this technology could, be integrated into your biomanufacturing process. Looking back, I'm just like, that is just insane. But I was, going through this process, and we actually had 3 large pharmas that got back to us and had some initial conversations, and they gave us those initial specs on, hey, if you can actually achieve these areas, within your technology, we'd be interested in a partnership. And so we used that to get our initial angel round, to refine the technology to ultimately meet these specs. We ended up going back to one of them being a large pharma, Eli Lilly, and showed them the data, and they got really excited about what we were doing. They're like, let's do a proof of concept. Let's see if you can produce five of our proteins that kind of represent the different scaffolds or therapeutic modalities that we're producing at Lilly, and see how well the technology actually works with the products that we have. And we ended up being successful with all 5 of those molecules, and actually gave us an acquisition offer at that point in time. And I remember going to my dad and telling him, we're turning this offer down. We have big aspirations. Like, this shouldn't be just for one pharma company. This needs to be a platform that every single company can utilize, and ultimately helping reduce the costs for patients. And so ended up using that to raise our Series A financing. And there's a lot of ups and downs, a lot of grit and determination. And I think throughout this period is where I really realized that you as an entrepreneur, you have to push yourself outside of your comfort zone. You have to be able to go and literally knock on those doors and ask for mentorship, ask for funding, ask for partnerships, and continually to learn along the way. And kind of the next journey of the entrepreneurial journey for me was realizing that all entrepreneurs end up getting lucky. And when you have luck, you have to be able to go capitalize on it. And we ended up having a really interesting technology that emerged through luck. And this was our ACE screening assay. So we developed this technology where we could screen millions of different E. coli strains, looking at how well they could produce a particular protein of interest, looking at the quality and the overall yields or titers. And we ended up finding out that there was a mutation that had occurred in one of the antibodies that we were producing, which was telling us that we had a higher titer, but we went and purified each of those, and they ended up being the same titer. And we're like, what's going on? Well, we ended up going back to the sequencing and seeing that there was a point mutation, and that point mutation actually was giving us a higher affinity. And so we realized that this ACE screening assay not only could look at the developability or manufacturability and the titer, but it could also look at, or it could also, you could also see the affinity or the functionality of the antibody. And this is that kind of aha moment of like, oh, wow, you can now do simultaneous drug discovery with cell line development with this technology. And the data that we were able to generate was orders of magnitude more than any technology could at the time in terms of the affinities that we could get. I mean, normally you have to produce every single antibody in a CHO cell that you want to produce, and then go screen it on, let's say, a BLI or SPR. And you can only screen thousands of antibodies at a given time. This allowed you to screen millions, tens of millions of antibodies, looking at their functionality and developability. And that was right around the time transformers were taking off at Google in 2018. And it was this idea that we had of like, wow, if you could take this data along with these generative AI models, you could really go from this paradigm of searching for a needle in the haystack to actually creating the needle and being able to search a much larger search space and hone in on those exact attributes that you wanted the first go around, finding out where the antibody, being able to pick the epitope you want, have the affinity, essentially the functionality, being able to hone in on the developability and manufacturability. And that's then when we went out and acquired Denovium. This was a do we build it or buy it moment in the company's history. And we knew that we wanted to accelerate our growth. And so we ended up finding a company that we really believed in the people, the technology, and the original kind of deep learning models that they were developing. And that was Denovium. We ended up acquiring Denovium, and it's been, we've been integrating that platform over the last 4 years. And ultimately, that's allowed us to make the major breakthroughs in generative AI that we've had. If you look at ChatGPT or these large language models, right now what we're seeing is that these are compute limited. Like you're training on the whole internet. But if you look at biology, we have a lot less data to ultimately train on. And in biology, we're really data limited, not compute limited. And this ability to integrate the wet lab and the AI has really what's led to the tremendous growth that we've had over the last 4 years. And it's seen through our IPO that we did, our partnerships with Merck, with AstraZeneca, with Almirall, and also has led to the additional follow-on financing we just raised this year of $86 million. It's also allowed us to bring in expertise like Andreas Busch, who is the head of R&D at Bayer, as well as the CSO at Shire, to help utilize this technology to build out our own pipeline. And so you can see this journey. It wasn't a straight line. There's a lot of twists and turns, a lot of failures along the way. And Elon Musk said it the best. Being an entrepreneur is like chewing glass and looking into the abyss. And that's what it really does feel like. You have to have a high pain tolerance. If we didn't make the pivots along the way that we made, and we didn't double down or triple down in some of these areas, we would have been out of business. If we would have still stayed at cell line development, we wouldn't be in business today. And I think it's this journey really does show that you have to have you have to be very, very passionate about what you're doing. You have to have a high pain tolerance because there are a lot of failures along the way. And you have to be willing to make the pivots and the changes necessary in order to continue to evolve and stay alive. I mean, it's to me, this is like evolution at its finest. You're adapting and you're growing, and it's and it really is. Actually, last night we had dinner, and was asked by Tina, Bob's wife, like, do you have any regrets along the way? And I said, absolutely not. Like, I've had a lot of mistakes, had a lot of failures, but they've all been learnings. They've allowed me to grow as a CEO, as a leader, as an entrepreneur, and it's allowed the company to be where it's at today and be truly on the forefront of AI drug creation and ultimately ushering in a new future, which I truly believe in the next 10 years, what we're going to see is the enablement of personalized medicine, where you can take a patient sample, find the target that's relevant for that disease, and then design the biologic that ultimately treats that particular disease for that individual patient. All right, I'll end with this. At the end of the day, it's all about team. We have an incredible team of unlimiters at Absci, and we'll dive into more of that. But I just want to first just thank our team. We wouldn't be where we're at if it wasn't for the incredible unlimiters at Absci. And so with that, I'll end it and turn it over to Bob.
All right, we're gonna move over here. This thing's still on or not? It is still on. And microphones will be coming around for people to ask questions. Thanks, Sean. That was awesome. I'll start with a question. How do you choose the targets? And I can imagine choosing targets that end up being a rabbit hole and a lot of time, and maybe not success at the end. I don't know if that's true. Maybe everything you've done has been successful so far, but how do you choose them? And are all the targets you're currently working on partnerships with pharma, or do you have some that you're within Absci choosing?
Yeah, absolutely. So we do have our own pipeline, and those are targets that we've chosen ourselves. And so the TL1A is actually one of those. And so we have what we'll call like a knowledge-based approach, which is leveraging the expertise of our team, like Andreas Busch and Christian Segman, that are able to look at and kind of have the knowledge of the biology that's already been explored and out there, and figuring out how you could leverage our platform to look at new novel biology that you couldn't have with previous technologies. And so this is kind of our best-in-class approach and what we've applied with TL1A. And then we also then have a more novel approach, which is our reverse immunology platform, where we're able to discover new novel targets. And what we're able to do with that is we essentially base this technology around tertiary lymphoid structures. And what's really interesting in tertiary lymphoid structures is that they have a very different B-cell antibody repertoire than what's in peripheral blood. And so we're taking these tertiary lymphoid structures from patients, mainly focused in oncology, and figuring out what antibodies are being made in these structures. And we then computationally reconstruct these antibodies, manufacture them, and then find out what antigen they're binding to, doing a proteome panel screen. And that's what's allowed us to discover ABS-301, which is a novel IO target and a novel mechanism. And one of the reasons that we got excited about this was one, it's kind of a new novel way of discovering targets, but from an AI approach, one of the big bottlenecks that we're seeing with this is actually the proteome panel screen. So actually producing these patients and then doing the panel screen. And so our thought was, like, if you could go in the reverse direction of our current AI model. So instead of going target to antibody, you could go antibody to target computationally. You could rapidly scale these hospital partnerships and rapidly de-orphan and discover new novel targets. And so we do have, again, a kind of a first-in-class target approach, as well as looking at targets that have already been discovered and seeing how we can leverage AI to create best-in-class assets.
Amazing. Yeah. All right.
Thank you for coming. You mentioned several times about your competitor molecules, biologics. And I was wondering, how do you know you're best in class, only in class, first in class? And what is your strategy when you think about your competition and how you position yourself to be better?
Yeah, absolutely. So, you have to look at, what ultimately is best for the patients. And we do think, going back to TL1A, being able to go from once monthly to once quarterly dosing, that's a huge convenience factor for patients. And especially with geriatric patients, we do believe that this will be great because you can tie it to patient visits to the doctor. We do believe that that is going to be an ultimate benefit to patients. And then additionally, with the kind of the monomer trimer binding and the potential to unlock new patient populations, that also is a huge advantage, and ultimately, allows you to differentiate in the market compared to the other competitor molecules. And so it's really looking at the convenience and potential efficacy. And so for best in class, we really do want to see a step change function when going after some of these known targets. But again, just looking at seeing if we can expand patient populations or increase overall convenience.
Thank you for your talk. This is my first attendance of an entrepreneurship talk. I was wondering, as the company grows, are there plans for shareholder dividends?
So ultimately, we do not plan on having any dividends for shareholders. I think what we are trying to do is have stock price appreciation, and so the overall value to shareholders is seeing the stock increase. And how do we increase the stock price? Well, we have clinical readouts that end up looking great, and we're able to monetize these overall assets, and that's what's going to end up causing stock price appreciation. So we're more focused on that than looking at shareholder dividends at this point in time. And also too, we aren't a profitable company, and so dividends also don't make sense when you're not profitable.
Hello. This is very exciting work, and just like I am starting a company that performs very similar functions to yours, so hearing the story is really interesting. I wanted to ask how you went about going from first turning down being bought out to identifying a company to buy, and how you identified Denovium as the ideal partner going forward, shifting into like the AI space.
Yeah, absolutely. So when we looked at purchasing Denovium, it really was twofold. One was the team and the talent. It was a small team, and we really needed to make sure that they could integrate well with our team, and the teams could really jive well together. And they wanted to continue to build the company together. Like if we ended up buying them and they ended up leaving, that would be a huge risk to Absci. And so I think it was making sure that the alignment was there, that we were aligned on the vision, and then also the technology. Even though the technology was I would say early in its kind of infancy and its build out, we saw some of the initial kind of proof points working, and we saw how it could integrate in with our wet lab technology. And so we saw all the synergies, and also to the you factor in the prices as well. And so all of those factored into the overall acquisition, and also to our former CTO had worked with these individuals before and could vouch for them. And so but the integration and making sure that everyone's aligned is super important, especially when you're doing a technology acquisition or an acqui-hire versus buying a bolt-on that you're looking to expand your revenue base. And so I think kind of two separate different types of acquisitions. And this one was definitely focused more on the team and the vision and making sure that everyone was aligned.
Your talk was really inspiring as someone who's trying to start a company, a biotech company here in Oregon. And obviously the landscape was very different in 2011. But if you were starting again in the basement lab in Oregon, what would you do differently with finding pharma partners and trying to raise money?
You know, it's interesting. I mean, I don't think I would have changed anything. I think the temptation that I would have had now would have been just to move to the Bay Area or Boston, because there's a lot more capital available there. I think early stage capital, call it, pre-seed and seed, there's definitely capital here, but to, get venture financing, it's definitely a more difficult environment. And that's something that we, as a region, that I'm really focused in on, is how can we continue to get more venture capital dollars here? Because that can really stagnate your growth if you're not able to tap into those particular pockets of capital. But we were able to do it. We've been extremely successful. Honestly, I thought we were going to move to the Bay Area or Boston at some point in time. This was just kind of a cheap way to get off the ground. I could move in with my parents, and is a very cost-effective way of starting a company. And when we had the capital,
Get some equity for that, or?
my dad actually did. He got 4% equity for a $40,000 investment. Yeah, he's
Can't still get that deal?
no.
It's a really, really interesting talk, Sean. Thank you for that. And my question is, I'm guessing that you have already moved into the market for bispecifics as well, and for other kind of antibodies. What kind of technological differences have you seen in terms of production of antibodies or versus production of bispecific antibodies or something in your engineered E. coli platform? How have you navigated through it?
Yeah, absolutely. So that was actually one of the niches that we focused in on originally with kind of the biomanufacturing platform, and was the basically everything kind of non-antibody related. So focusing on the BiTEs, the FC fusions, the bispecifics, and or even the Fabs. Those are a lot more difficult to produce in CHO cells. And so our platform is extremely robust for producing kind of these next generation antibodies. And so we can easily adapt our screening platform to screen for these. And we definitely have partnerships that are around some of these new novel modalities. And so, yeah, we're definitely set up extremely well to be able to screen, validate, and train our models on some of these novel modalities.
Sean, that was really inspiring just to watch that whole progress, and then the hockey stick at the end, right? So that's overnight success that started 10 years ago in some ways. So one
overnight success, and then it went down to, and we're working our way back up.
So one thing that I'm really taking away from this in terms of you're owning the whole life cycle of the AI process, right? A lot of places are employing models, but they're getting data from other places, and you own it from start to finish. And I think that's really critical of your vision. So I got two related questions related to that. One is the validation stage, or you've got your training data and then you've got your test data. That's basically what you're doing, that whole cycle. Yeah. So what are the metrics on the validation now? Yeah. And a related question that I think is a important one for this group is, so if you're looking out 5 to 10 years, what sort of engineering solutions to really improve that test data acquisition and training and test would be critical for a phase shift in your company?
Yeah, absolutely. So it looks like we're owning the whole AI process, but really what we've done is actually we've focused on a particular niche that we believe is going to unlock a ton of value, and what we believe that is the ability to target any epitope that you'd like, or being able to do epitope landscaping. So let's say you have a brand new novel target. You have no idea what epitope that surface exposed is going to give you that biology. You can now have a platform that can generate antibodies that bind to all those different epitopes of interest, and then go into the wet lab and screen them in vitro assays and see which of these cell-based or which of these antibodies gives you the biology that you're looking for. And I think that that's a huge breakthrough in the industry, but it's a breakthrough in the design of antibodies. It's allowing you to test hypotheses that you haven't been able to test before, but it's not predicting the biology. And that's where I want to go. I want to figure out how can you actually start using generative AI to predict what targets you should be going after, what epitope you should be ultimately looking at, and then ensuring that then can that translates into not only in vitro assays but translates into in vivo assays. And it's we were talking about this last night. I mean, it's kind of like a systems biology screening approach, and it starts to get more and more complex. I mean, design is a much simpler task because we figured out how to unlock some data to do that. But it gets a lot more complex when you're looking at a whole system, when you're looking at the whole biology altogether. And I think that that's like the next iteration of Absci, is being able to kind of start to predict some of that biology and that translatability. And I think that that's a really exciting new frontier that I think will be explored in the next 5+ years.
Thanks so much for joining us today. I work with academic startups, and in asset development in the biopharma space, they often say, I'm going to take this all the way to the market. I heard you mention how even where you all stand today, you work very closely with partners and at later stage clinical trials, work with them to then finish, I would assume, phase 3 and then launch. Could you walk us through your philosophy and how it may have evolved in the 10 years of the company as well?
Absolutely. So we originally focused, even in the early stages, it was all platform focused. So it was partnering and then ultimately licensing out what we had, either whether it was the E. coli strain or the asset that we had developed. And we ended up pivoting into a hybrid business model where we're developing our own assets, but it's still, at the end of the day, is partnership driven. We don't plan on taking any of our own assets past phase 2 proof of concept. Because if you look at the value inflection points, there's a few along the way. There's the first value inflection point that you have when you can sell an asset is when you have in vivo proof of concept. That's usually a typical stage where you go raise a larger Series A. And pharma also, if it's a new novel target, would be interested in acquiring that. Depending on the indication, you can have your phase 1 that generates potentially efficacy signals. And based on those signals, that's a point in time to go and either raise additional capital or sell that asset. And then the biggest value inflection point in pharma is the phase 2 proof of concept. And that's where you're going to be able to generate the most value for a particular asset. And that's the value inflection point which we will stop at. There are some biotechs that plan to commercialize assets themselves, but what we've realized and seen is that pharma is great at that. Pharma is so good at late stage clinical development, the manufacturing, the developability, and so put it in the hands, put it into capable hands, and take that in investment that you just or take the cash that you got from the acquisition and invest it back into developing more assets. And so that's how we see value generation. And so at the end of the day, it's actually still partnership. It's just a question of, do you partner at the target stage or do you partner at a phase 2 proof of concept? And so again, you have to map out these different value inflection points. And I think that's what investors want to see when you're going out and fundraising. It's like, okay, I need to raise this amount of capital, and this amount of capital will get me to a value inflection point, which will then increase the overall value, which then allows me to go out and raise more capital. But as an entrepreneur, you always have to be looking at what are my value inflection points and how do I ensure that the capital I raise absolutely ensures that I can get to those inflection points.
Great talk. I just want to get at a little bit more of the science side. So, what was kind of like how you like landed specifically on choosing the antibody protein scaffold? And in the future, do you plan to look at other like protein structure scaffolds for developing therapeutics?
Yeah, absolutely. So our pipeline right now is mainly focused on antibodies, but I think one area that we see as actually two areas that we see as really exciting from a modality standpoint is bispecifics and additionally ADCs. And so, you know, we do have partnerships with AstraZeneca that are using this platform to develop the initial antibody, which then they're going to use their ADC platform to create some novel ADC assets. And so those are definitely kind of two areas that we're going to be expanding into in the future.
Bob?
I enjoyed hearing you refer to your team as unlimiters, and I was just wanting to hear your thoughts on that.
Yeah, absolutely. So in order to be able to actually see a vision through and accomplish big, audacious like goals, you actually have to believe that you are going to achieve it. I think a lot of times in science, you know, science is like difficult. Like it's you can get, you know, beat down, you can get jaded. You know, this is never going to work. And if you actually believe that it won't work, you're never actually going to go and solve it. And so we have a value at Absci, which is believe in the impossible. And it's the belief that, you know, we can achieve it. Like from a, if it is possible from a first principles perspective to achieve it, then let's figure out how much, you know, time it's going to cost us to figure it out, and you know, what's the overall cost. And so it's, you know, ultimately time and cost, and that you have to factor in. But I think it's making sure that everyone does fundamentally believe that we can get there. And so you know, it's really about this idea of turning what is impossible into possible every day.
Well, I think that's a really inspiring way to end, a little insight into your mindset, which is really cool. It's awesome. So Sean, thanks so much for being here today. Thank you all for coming. We're going to take a break with the Entrepreneurship Speaker Series for the summer, but we'll be back in the fall. I also want to say thanks to the staff who've been supporting this series all year long. So Sean, that was an awesome talk. Thanks so much.
Yeah, thank you, Tom.